Okay, we will go ahead and get going as people continue to trickle in. Welcome, everyone. Today's talk is entitled Maximizing 3D Muscle Tissue Data and Minimizing Cell Usage with Mantarray Mini Plates. My name is Ari, and I'll be helping guide us through this event. The webinar is designed to be interactive, so please, if you'd like to go ahead and let us know in the chat where in the world you're tuning in from right now, that would be a wonderful start. Additionally, the webinar does include a Q&A session, so please feel free to submit questions for our speakers anytime throughout the presentation. For those unfamiliar, you can find the Q&A and chat functions at the bottom of your Zoom screen. We'll be attending to the questions after the main presentation. Now, it is my pleasure to introduce our speakers for this event, Dr. Christos Michas and Dr. Shawn Luttrell. To give a brief background, Dr. Michas is a product manager at Curi Bio, ensuring that the company's cutting-edge research translates into impactful products. He holds a PhD in biomedical engineering from Boston University while working with Professor Chris Chen, where he developed 3D microphysiological models of cardiac failure. Christos holds various patent and top-tier journal publications under his name and has a keen interest in the commercialization of innovative science. Dr. Luttrell is a product manager at Curi Bio. She graduated from the University of Washington with a degree in biochemistry and molecular biology, followed by graduate studies in developmental biology. She completed a postdoctoral fellowship at the Institute for Stem Cell and Regenerative Medicine at the University of Washington, where her focus was on stem cell and gene therapy treatments for neuromuscular diseases with an emphasis on iPSC-derived muscle development. Okay, without further ado, I'd like to hand over the presentation to our speakers. Christos, please begin whenever you are ready.
Thank you very much for the introduction, Ari. I'm Christos, and here with Shawn, we will be telling you about two new advancements that we have made within Curi Bio. The first half of the presentation will be about the Mantarray Mini Plate that I will present. And then Shawn in the second half will tell you about the cardiac media that we have recently developed. I will begin the presentation by telling you a little bit more about Curi Bio. But before I do that, I would like to ask you to tell us about yourself. So in the next minute, a pop-up window will show up on your screens. And please take the time to answer the 3 questions that are in that pop-up window. The first question is, what applications are you interested in? So early discovery, target validation, toxicity screening, efficacy or potency, basic research, or other. The second is whether you have experience with the 3D tissue technology, yes, no, or not applicable. And then finally, the last question, which of the following have been a barrier to adopting or scaling 3D tissue work? The answers being the technology is too expensive, you don't have the right resources, as in the right cell lines, the right media. You would like to have more validation in the technology doesn't scale. You need a solution with regulatory endorsement, or casting and experiment should be more automated. So please let us know. You still have 20 to 30 seconds. Nice. Answers are rolling in. And we have quite a spread. 10 more seconds for you. Great, thank you very much. We'll go ahead and end the poll now. It really seems that you're spread across the entire spectrum in all questions, so we really appreciate that. So let's talk about Curi Bio. So Curi Bio helps you make your R&D decisions from early discovery all the way to IND by integrating human iPSC-derived cell models, tissue-specific biosystems, and machine learning-enabled data analysis. And we do this for discovery, for disease modeling, for safety and toxicity, and efficacy and potency. We also do this in two ways. You can interact with us in the form of services. So you come to us with your ideal study, and then we help you identify the resources you need for that, perhaps build those resources, design the experiments, run the experiments with our technology, process data, and then reach conclusions so you can have everything to make your decision. We also do this in the form of products, and this is what we're going to focus on in this presentation a little bit more. We have cell, media, and consumables that help you build 2D and, most importantly, 3D cultures. We have systems, or biosystems as we call them, so you can run experiments with these cultures and extract data. And finally, we have data analytics tools, so you can process the data and reach the conclusions that you would like in the convenience of your own lab. And starting, we're going to go into the Mantarray plate. So this is a core consumable that Curi Bio provides, so you can use 3D tissue technology. What you're seeing in the photo over here is the Mantarray plate that is a 24-well plate, and within each one of the wells, you see that little strip. So that is a tissue that is suspended between two posts, and it can contract. And so we have built this because we believe in the benefits of 3D tissue technology. Specifically with 3D tissues, you have improved physiological relevance because you now have a 3D structure with an extracellular matrix that embeds chemical and mechanical cues and also includes multiple cell types that can interact with each other. Also, compared to 2D cell cultures, you have extended culture times, so you can go up to multiple months of cell culture. And this is important for building elaborate experiments that can also reveal more latent effects. So for example, for a typical gene therapy experiment, which is amenable with this platform as well, when you add your therapeutic, it might take up to 60 days to see an effect. And that effect is not just gene expression, it could also be the full-fledged effect you'd like to see in the clinic, such as function. And so that includes contractile force, the time kinetics of that force. And that is a third point that's being made, that with this technology, you have access to this function. And so what Curi Bio has done is not truly build from scratch the 3D tissue technology, but pioneered simplifying the tissue creation and maintenance of this technology so you can use it in your lab. And so when you use a Mantarray plate, you will see that you get over 95 casting success rate. And this is enabled by the structure of the consumable. So if you look at the Mantarray plate, you will notice that there is a Mantarray lattice. So that is a set of 24 pairs of posts from which the tissues are suspended. And with the Mantarray plate as well, you're going to get the casting plate, which helps you cast or create the tissues. If you were to place a Mantarray lattice and the casting plate together, you would get a situation like in the image on the top right, where the tips of the posts are situated within a very narrow well. And so all you have to do is go in with your pipette or your automated robot and extrude hydrogel and cells, which are then going to compact and spontaneously form tissues. Once the tissues are formed, all you have to do is lift the Mantarray lattice and place it within a Mantarray culture plate in fresh media. And as you maintain the tissues, all you have to do is switch out the culture plate again. So now that I have showed you how to make tissues and how Curi Bio has simplified that process, what do you do with them? So first, you might want to paste them or condition them. And for that, you have the Stingray stimulation system. So this is an instrument the size of a shoebox. It can optionally be placed in your incubator if you choose so. It has a very simple, intuitive, user-friendly software that you can use on your computer that is connected to the Stingray. And then using the stimulation lid, which you simply slide into the plate, you can stimulate all 24 tissues. And in fact, you can apply a different protocol to each one of the tissues, so you have individually addressable electrical stimulation. If pacing and condition is not enough for you, and you also want to get contractile data, that's where we have the Mantarray contractility system. So before I explain the system, let's look at the consumable on the left. So you see the green circle. So at the very tip of the white flexible post, there is a black dot. And that black dot is a magnet. And when the tissue contracts, it moves the magnet. And so if we were to take the entire plate and place it on the Mantarray instrument, which you're seeing in the middle, the Mantarray instrument is equipped with magnetic sensors that can track the position of the magnets, all 24 of them, and in real time for all 24 tissues, present to you the contractile force of the tissues. This is still small. It is a shoebox size, can go into the incubator, has a software, so it's intuitive and user-friendly, and it is compatible with the stimulate, so you can also apply stimulation at the same time. So as you're getting the data on the right here, the next question is, how do you use the data? How do you process it? And the answer is very simple. The data is automatically uploaded to the Pulse Cloud platform that Curi Bio provides. So each one of those contractions is automatically broken down into multiple points, as you're seeing on the bottom right. And then from there on, those are used to calculate a host of metrics that you can use so you can plot your data and make your conclusions. In this plot over here, for example, we're using the active twitch force to tell apart different conditions. If contractility is not enough for you and you'd like to expand further to other types of outputs, then we also have the Nautilite electrophysiology system. This is for optical mapping of voltage in calcium. It has a very high resolution and it is compatible with 2D culture plates up to 384 volt plates, and then with the Mantarray 3D tissue culture consumables. And again, it has its own software and it has a range of applications that you could use it for, including cardiotoxicity screening, electrophysiology, and calcium transients. And so with this, I took a few minutes to present to you the tool belt, the tools in the Curi Bio tool belt that you can access. But as I said, part of the mission of Curi Bio, excuse me, is to make this technology accessible. And so looking at these products, what can we change? So we're going to focus on the Mantarray plate here. And let us say we run a typical experiment. So we're going to cast 24 skeletal muscle tissues in a Mantarray plate. We're going to do this at day 0. Then we're going to wait for 10 days so the tissues form fully fused myotubes. And then we're going to keep the tissues for an additional 20 days to run our experiments. If we were to break down the costs of that experiment, you would notice that the vast majority of your dollars go to cells. And even if financial resources are not your constraint here, we asked you, and you did tell us, that cell culture can be time-consuming and you'd rather avoid it, that some cell lines are just not as available, such as primary cell lines. And then the other thing to note is that our platform is well suited for therapeutic modalities that are very complex, including viral therapeutics. And the thing with viral therapeutics is that their load scales linearly with cells. So all of these are incentives to reduce the number of cells in the tissue, or in other words, to reduce the size of the tissue. Therefore, Curi Bio is introducing the Mantarray Mini Plate. By reducing the post distance by 30% and reducing also the volume of the casting well, you go from tissues that look like this in the Mantarray plate to this in the Mantarray Mini Plate. And so similar shape, but smaller size. And thanks to the small size, you gain access to several benefits, including up to 45% reduction of the cell count, 60% reduction of the casting volume, and 40% savings. And note that for the experiment that I described to you previously, 40% means approximately $2,000 per plate. And all of these benefits come while retaining the over 95% casting success rate. If you take a moment and look at the numbers more carefully, you will notice that the cell count reduction is a little bit smaller than the casting volume reduction. And so that effectively means that you're going to end up with denser tissues that are one step closer to replicating a tighter sensation between the myotubes. So on the left, you have the skeletal muscle in the Mantarray plate, and on the right, a skeletal muscle in the Mantarray Mini Plate. And you see that the green dots, which stand for myosin heavy chain, and therefore the myotubes, are much denser in the Mantarray Mini Plate. Now that we know that we can make the tissues, and in fact, they have improved density, how compatible are they with the existing infrastructure and can we use it with it? And by that, I mean the Curi Bio instruments that I mentioned previously. So starting off with the Mantarray contractility instruments, we're going to use this with the engineered skeletal muscle tissues. And so the experiment we're going to run, we're going to cast skeletal muscle tissues at the standard cell count in a Mantarray plate that is 100%. And then in a Mantarray Mini Plate, we're going to use a scaling a range of cell counts from 24% to 104%. So increasing cell count, increasing density. And as we increase the cell count, you see that the forces generated by the tissues also increase to the point that they match or even exceed the Mantarray plate. These specific numbers will vary depending on the different cell lines you use, perhaps the media, the cell ratios, etc. But Curi Bio has identified that 56% is a good reduction. So you have sufficient savings, but also high quality data for all the different types of tissues you might make. When it comes to cardiac tissue, we've noticed again that 50% or in sometimes 70% is sufficient to match the quality of the data of the Mantarray plate. The second platform that we're going to use is, of course, the Nautilus platform for electrophysiology. And so again, we're going to take the same plate with the variable cell count, Mantarray Mini and Mantarray, and place it on the Nautilus to observe the calcium transients produced by these tissues. And so what is important to point out here in this plot is that for all the different conditions, you get excellent signal to noise ratio, and in fact sufficient so you can run all the studies you would run with the Mantarray plate on the Nautilus. Furthermore, if we take the 56% tissue and we subject it sequentially to different stimulation frequencies with the stimulant, and we're just presenting them here overlaid for your convenience. So we see that we're driving the tissue from individual twitch contractions at 1 hertz to almost fully fused tetanus at 50 hertz. And in fact, the fluorescence resolution and the time resolution are high enough for you to be able to notice the very fine oscillations that happen at the very top of the 50 hertz, almost fully fused tetanus. And so that is to say that with the Mantarray Mini and with the Nautilus, you can get really robust electrophysiology data. Last but not least, the Mantarray Mini retains compatibility with all other standard assays. That includes media assays and cell assays. As an example here in the plot on the top right, we present how much cardiac troponin I is secreted by Mantarray Mini tissues and identified with ELISA in the media. And so you see that the amounts are high enough for ELISA to be able to distinguish not just to conditions, but also to different phenotypes, healthy and disease. And when it comes to other assays, the Mantarray Minis do produce enough material so you can run them. So for genomic DNA, for RNA sequencing, protein quantification with the Western blot and GES, and of course with staining, as I showed you previously. Apart from the fact that the Mantarray Mini retains all the benefits and adds a little bit of improvements, it also gives you access to new opportunities. And a good example of that is the neuromuscular junction model that we created at Curi Bio. This is a new model, and it is a 3D functional model. So what we have done, we have co-cultured neurospheres with skeletal muscle mini tissues. And what happens is that the neurospheres grow out axons into the muscle to innervate the myotubes. And in a fashion that if we stimulate selectively the neurospheres, then we see a contraction in the muscle. And so that reveals the presence of a neuromuscular junction. And in fact, the neuromuscular junction is inhibited if we add Botox. And furthermore, we can quantify the effect of Botox in a similar fashion as the mouse lethality bioassay to quantify the Botox potency. And you see that on the bottom left here. If you're interested to know more about this new assay, and if you want to work with us to help develop it further, then please do look at our application notes that are on our website, and also the webinar that can be found on our website. In summary, I presented you the Mantarray Mini Plate, and I told you that the Mantarray Mini Plate retains all the amazing benefits of the Mantarray Plate, that is high casting success rate, stimulation with a stimulant, functional data with the Mantarray system, electrophysiology with the Nautilus system, and compatibility with standard media and cell assays. And beyond that, you also get improved cell density, substantial savings, and new applications as I presented the neuromuscular junction model that we created. So now that we have this additional tool in our toolbelt, the Curi Bio toolbelt, a question you might be asking is, how can you run a comprehensive study with all these tools that you have? And so this is a great segue to go into the second segment of the presentation, where we're going to use this same technology to develop cardiac media for 3D tissue cultures. Shawn will tell you all about that. And as we proceed to the second half, I do have a question for you and regarding media. So another question will pop up on your windows, and we would like to know, what cardiac media are you currently using for 3D in vitro culture? Is it RPMI-based formula from the literature? Is it a formula that you developed, some commercial source, or single cell something else? Let us know, and as you're answering the next minute, I will hand off the presentation to Shawn.
Hello, everyone. So it looks like answers are still coming in. We'll give this another 20 seconds or so. So it does look like answers are a bit spread between the different groups, but a lot of you are using the RPMI-based formula for your cardiac tissue. So that is really nice, because it is in line with the data that we're going to be showing you in this next part of the presentation. Okay, so let's go ahead and end this. And thank you, Christos, for that nice overview of the Mantarray Mini technology and our biosystems that we use to measure function from those tissues. So I am Shawn Luttrell. I'm product manager for cell and tissue models. And today I'm going to talk about the kinds of data we can get from the mini-sized tissues, with an emphasis on the mini-engineered heart tissues, or EHTs, as we like to call them. And so the first step in generating our mini tissues is differentiating or acquiring the cells. iPSC-derived cardiomyocytes have been around for quite some time, and the differentiation protocols are fairly robust and easy to use. As such, stem cell-derived cardiomyocytes are becoming more popular for in vitro assays for a variety of reasons. You can generate a virtually unlimited number of cardios from the iPSCs. They allow you to recapitulate a patient's disease in a dish or model a new disease. You can edit the cell lines to make isogenic pairs, which can be used to model any disease with a genetic foundation. So stem cell-derived cardiomyocytes have really become a test bed to study cardiac biology and different therapeutics aimed at treating cardiomyopathies and other diseases. However, the challenges in working with these cardiomyocytes have historically been the immature state of the cells and the difficulty to model some diseases, disease phenotypes that have a late onset or present with maturation. Another obstacle can be the purity of the cardiomyocytes after differentiation. So when we make our mini-engineered heart tissues, we want the majority of those cells to be cardiomyocytes. Additionally, we want to ensure that the non-cardiac cells are minimized to limit the number of unknown cell types that we might be introducing into our heart constructs. And finally, batch-to-batch variation, so maintaining consistency across multiple differentiations to help ensure consistent experimental results over time. I'd say now, with improved differentiation protocols and cell enrichment methods, purity and variation concerns have been somewhat mitigated in recent years. But the question of maturation, however, is still a big hurdle in iPSC-derived cardiomyocytes for preclinical testing. And so currently there are several approaches that have been used to enhance the maturity of these cells in vitro. Some of these include aligning the syncytium of cardiomyocytes and stromal cells to produce ordered development of the muscle, synchronous electromechanical coupling of contractility, supportive extracellular matrix and scaffolding structure to mimic the mechanical and biochemical properties of native tissue, and also functional microvasculature to provide adequate nutrient and oxygen delivery to the tissues. So many of these maturation strategies are not easily achievable in traditional platforms. And that's really what motivated us to work on developing our advanced engineered heart tissue platform. So this could allow us to integrate complex maturation factors while ensuring reproducibility across tissues. And so the mini heart tissues and Mantarray combine multiple maturation cues to improve functional outcome for disease modeling and therapeutic testing. So the first step in building our mini engineered heart tissues is making the cells. We use a small molecule directed differentiation approach to generate our cardiomyocytes. This has been well described in the literature. We start with iPSCs, and by day 8 to 10, we have a synchronously beating cardiac monolayer. We enrich these cells to increase the purity, and we confirm that purity by using flow to test for cardiac troponin I. After a couple of weeks, we have a fairly pure population of cardios that we can bank in high-density cryovials for future use. So then when we are ready to make our tissues, we can thaw a single cryovial and cast those cells directly into our mini casting plate for each study. And since we're using the same lot of cells over mini plates, this improves the consistency and the repeatability between tissues and between different experiments. So once we have our cells, we can make our 3D engineered mini heart tissues. So these tissues will start to spontaneously beat after 2 to 4 days, and then we can measure function in a variety of ways across our various biosystems. We can use the Mantarray pictured on the left to measure basic contractility over time, looking at force and kinetics. And after we achieve a stable beat rate and contractile force, we can apply drugs and different compounds to look at inotropic responses. We can use our Nautilite platform to measure calcium handling dynamics in the tissues over time, and that could change as these tissues develop and hopefully mature in our 3D culture. And then we can measure more complex biology like force-frequency relationships and post-rest potentiation, which may speak to the level of maturation that we might be achieving in our muscle constructs. So in evaluating our mini tissues across our platforms, we look at a variety of contractile metrics and functional outputs. So we start with morphology, looking at things like cell viability and tissue compaction. So at the top of the slide, you can see a full plate of our mini EHTs. We ensure these tissues are uniform and consistent across our different groups. After they start to beat, then we can measure basic contractility. We can measure force, beat rate, kinetics, any contractile irregularities that might be happening in the tissues. And once they are stable, then we can assess some more complex biology like calcium handling, response to inotropes, positive force frequency, disease phenotypes, things like that. So in developing our 3D muscle tissue platform, one thing that we found was that media really matters. What you feed these tissues makes a big difference in the functional output that you get. And we originally developed our platform using RPMI plus B27 plus insulin, which a lot of you are currently using today. This is a well-described cardiac media that's been in the literature for quite a while to differentiate the cells and keep them in culture. And it does work to produce functional tissues in our system. However, what we found, though, is these tissues are fairly weak little muscles that don't achieve the hallmarks that we would expect from a more mature tissue. So things like positive force frequency relationships, positive post-rest potentiation, and importantly, for many of our studies, expected drug and therapeutic responses. So this is really what motivated us to work on a cardiac media that helps us achieve these things. And we used our mini tissues to help lower the cell resources over the last year while we worked on developing this formula. So the rest of my slides, I will show you the outcome from these experiments, which are all done in the mini engineered heart tissues. Okay, so to start, we looked at basic contractility, force over time. And in this graph, we have a contractile waveform that's recorded on the Mantarray. We have force on the Y and time on the X, and we can really pull a lot of contractility data from this one waveform. Here I'll show you a graphical representation of how we measure the contractile metrics from each beat in this waveform. So moving from the baseline to contraction 10, from there to the peak height, that is our twitch force. And we can plot the mean of all of these contractions to get our force for this particular day in culture. When we plot these forces over time, we can get a graph like this. And this is where we can see how the tissues are developing throughout our experiment. So here we have 3 different cell lines, 1 internal patient line and 2 commercial lines. We culture these tissues in either RPMI plus B27 plus insulin, which are the graphs in red and orange, or we used our cardiac media graphed in blue and green. And I think it's clear to see tissues cultured in RPMI are much weaker and they drop in force quite early. Whereas our physiologically relevant media, on the other hand, with many molecules at similar concentrations to in vivo levels, is producing tissues with a dramatic improvement in force and longevity when we compare this to RPMI. And so I'm only showing you through day 21 here, but these tissues can last for months in culture. They will last about as long as you can keep them sterile. So I'm only showing you through day 20 because we use these tissues for drug studies that I'll be talking about in a few slides. And so we can also see that results are fairly reproducible across these different cell lines. So when we plot our mean force over time in our muscle constructs, it doesn't really show us the whole picture. It doesn't show us if there was any irregularities in the individual contractions. And so by looking at the contractile waveform recorded on the Mantarray, we can see every beat of that muscle, and it can be very obvious to detect irregular patterns like this one that you see here. Now, this contractility can be caused by disease or arrhythmias in the muscle, but it can also be caused by phases of normal development and remodeling of the muscle tissue. So it can be a good indicator of how the muscle is developing and hopefully maturing over time, which is greatly influenced by the culture media. And so here we plotted the coefficient of variation in the twitch force across all 3 of our cell lines. We have tissues cultured in Curi Bio media in blue, and RPMI tissues are in green. Each dot on these graphs is an individual tissue. We typically want to be below 5% variation, indicating the tissues are producing consistent forces for each beat. And you can see when we culture the tissues in Curi Bio media, the forces stabilize within a couple of weeks and contractility becomes very consistent. It's interesting to know also that in the cell line 2, tissues were not even functional in RPMI media, so we couldn't even detect their contractility. Finally, we looked at the kinetics to ensure our model is contracting and relaxing at a consistent and healthy rate. At the top of the slide, you have examples of how we measure contraction 10, as well as 50 and 90% relaxation from these waveforms. So contractile kinetics of the tissue correlates with the calcium handling capabilities along with any irregularities. Slower kinetics may indicate compromised development of that contractile machinery. And what we can see is both contraction and relaxation of the tissues were physiologically relevant in our media, and they were faster in our media, suggesting a more healthy developmental state of the muscle. Next, we wanted to look at some compound responses. And one of the intrinsic functions of cardiomyocytes is they produce a positive force frequency response to increasing levels of external calcium. So we tested contractile response to various levels of external calcium concentrations and recorded that. We recorded that on the Mantarray. Twitch force, as you can see, increases with the increase of external calcium across all of the cell lines in our media. And it's quite dramatic, but it's fairly unresponsive in RPMI. And this may indicate impaired or reduced ion channels in these tissues. Next, we tested beta-adrenergic receptor against isoproterenol, or agonist, excuse me, for isoproterenol, which is another positive ionotrope. This is a widely used drug to treat bradycardia and heart failure. It positively regulates calcium handling to increase calcium in the cell, activating more cross bridges, producing a higher twitch force. And we see virtually no response or no positive increase in force in RPMI. And actually, there's a decrease in force at the highest concentrations. Whereas we see a very strong and clear positive response in tissues cultured in our media. And so we see that we can use these drugs to increase contractile force in our mini heart tissues. Our next question was, can we use drugs to decrease force in our tissues? And for this, we used a negative ionotrope for Apamil. This is an L-type calcium channel blocker. In vivo, it widens blood vessels and relaxes heart muscle. It's used to treat angina and high blood pressure, heart arrhythmias. When we apply this to our mini heart tissues, we can see a clear reduction in force at the highest concentrations in our media. However, tissues cultured in RPMI are either no response or an inconsistent response. And since this drug is an L-type calcium channel blocker, this could indicate tissues grown in RPMI may have reduced expression of this ion channel, which is in line with our previous calcium experiment that I just showed you. So these compound responses in both positive and negative ionotropes clearly show tissues cultured in Curi Bio medium are responding in a way consistent with improved calcium handling, which may indicate improved maturity of the muscle construct. A more definitive way to measure calcium in our 3D mini heart tissues is to use our Not Alive platform that Christos described earlier. So here we can load these tissues with a calcium indicator dye and then record fluorescence as calcium moves between the SR and the cytoplasm during contraction and relaxation. The Not Alive measures calcium flux in our tissues in a very similar way that we're measuring contractility on the Mantarray, except now we're measuring calcium movement in the cells through fluorescence. And so here we have a video of one plate of tissues. So this is one of the cell lines in our study, cell line 3. And we are culturing this plate in tyrode solution that contains a calcium concentration of 1.8 millimolar. That's physiologically relevant. Use this pointer here. In column 1, we have tissues that are grown in our Curi Bio medium. And in column 5, we have tissues in RPMI that were grown in RPMI medium. And you can easily see the tissue light up as the calcium moves in and out of the cytoplasm during contraction in column 1. But if you really squint your eyes, you can barely see movement in the RPMI tissues in column 5. It becomes more obvious, when we look at the representative traces from these wells. So we have in well A1, representative trace from Curi Bio medium tissues. And then in well C5, we're showing you a trace from the RPMI tissues. And it's really quite obvious. You can tell just by looking at these waveforms, there's a big difference in calcium movement, both in amplitude and duration. Looking at the change in fluorescence over the minimum fluorescence. When we quantify this across our 3 different cell lines in our study, we see a significant difference between the 2 media formulations in all groups. The fluorescence amplitude is much lower and the decay rate is significantly longer when tissues are cultured in RPMI. And so this is another good indicator that we're achieving improved maturation of the muscle tissues, as longer relaxation or decay time often indicates inefficient reuptake of calcium, which could relate to the immature state of the muscle, since these cell lines are all healthy and they're not diseased. And so with that, Excuse me. So with that, I'll recap by saying that at Curi Bio, we strive to integrate our biological models and platforms into one seamless workflow that provides our customers with everything they need to generate human relevant data for their research programs. Starting with the cells to make our various 3D tissues, media to support those tissues over time, biosystems to measure function from the constructs, and AI analytics platforms to generate meaningful insight from that data. Now at Curi Bio, we're proud to offer this new 3D cardiac tissue medium through an early access program to help drive your cardiac experiments forward. The media comes in growth and maintenance formulas to provide support for all days of your culture. I would also point out that 250 ml of this media feeds a 24-well plate of the mini or classic size tissues for up to 10 days. And so in summary, I think I've shown you today that we can generate human relevant data using our mini tissues, which come with a significant savings on cell resources. Our cardiac medium not only improves the contractility in our mini heart tissues, it produces tissues that deliver more reliable responses to compounds. And our overarching goal with all of this is to really generate human models, human in vitro models and systems that improve the predictive power of preclinical testing. And so how can you work with Curi Bio technologies? Well, we work with our customers in a variety of ways. You can purchase our instruments and our consumables outright. We work as a CRO executing service projects for our customers, and we can provide consulting services, or you can license some of our systems and models. With that, I will end and say thank you. And thank you for attending our webinar, and Christos and I are happy to answer any questions you might have.
Perfect. Thank you so much, Christos and Shawn, for those wonderful presentations. That does bring us to our Q&A portion. Everyone can continue sending in questions if you have any, but we have received a few already, so I'm going to start with those. Okay, I've already been casting with the large plates. How different is casting the mini tissues?
So I can answer that. The process is pretty similar. The only difference is changing the volumes that you add in the casting plate, and of course the size of the casting plate and the distance between the tissues. And so the protocols per se are almost identical. It's just a few numbers that change. Yeah, that's the answer.
Awesome. Thank you, Christos. Does your media contain serum?
I can answer that. So we have two flavors of our media. We have a growth formula for days 0 through 4, and we have a long-term maintenance formula for days 4+. The maintenance formula is serum-free. However, the growth formula does contain 10% serum, and what we found when we cast our cardiac tissues, if we can keep them in serum for just a few days after that initial casting, it really improves the development and the outcome and the resulting contractile force of the tissue. It basically acts to keep those cells happy as they're initially remodeling those tissue constructs and basically reaching a more steady state initially after coming from cryopreservation. That's another thing that we do, and I think I might have skipped over that during the presentation, but how we make our tissues, we take them from cryopreservation, thaw them, and put them directly into our muscle constructs. So with that, it's a pretty stressful event. So keeping them in a little bit of serum initially helps mitigate any negative response to that process. So yeah, maintenance formula is serum-free. Our day 0 through 4 formula does contain serum.
Awesome. Thank you, Shawn. Next question's from Juan. Is your 3D culture technology compatible with life imaging by fluorescent microscopy with high content microscopes such as Opera Phoenix Plus?
I can take this one. So I do not believe the Mantarray Mini Plates have been used specifically with the Opera Phoenix Plus. However, as long as the the focal depth of the system is sufficient, then we don't see a reason why it would not be compatible. And if you're unsure yourself, we can send you a consumable. We can send you so you can test it on in your lab. I will say that for the 2D consumables, which we did not talk about, so that's the nanosurface plates that help you have better alignment with the topographical cues on the bottom of the plate. Those have been tested with the Opera Phoenix, and those are fully compatible.
Okay, perfect. Thank you, Christos. Next question is from Gwendolyn. Have you looked at changes in differences between healthy and disease models in the standard versus mini plates?
I can take that. So we've started doing some of that work. That's a really great question. And initial data suggests that, we're still able to capture the disease phenotypes in our mini tissues, so there doesn't seem to be a difference. But I would say, disease modeling is something that we're just moving into with our media. So that was one thing that we really needed to develop to accurately model different disease states was the media. So the RPMI formula that we were using initially, we were not seeing the disease phenotypes that we would expect from different models that we use. Now we are seeing more of those phenotypes in our new improved media. So those studies, I would say, are just getting underway. And I think we'll have more of an answer to definitively show you that through the data here pretty soon.
Thank you, Shawn. Next question is, are there any drawbacks with switching to the minis? Excuse me.
So I would say there aren't per se necessarily drawbacks. What we know historically is that people would use a single tissue, so the same tissue, for multiple downstream assays, so for protein quantification and staining. And so with the Mantarray Minis, depending on your dexterity, you might not be able to do both with the same tissue. That is one. And so you can reserve the standard tissues for that. Beyond that, the Mantarray Mini should be able to perform what the standard tissues do. I would say that if you are a first-time user, it might be best if you start off with the large tissues first, so you can familiarize yourself with the casting process, and then you can move over to the Mantarray Minis. Yeah, otherwise, I wouldn't say there are any specific drawbacks of the Mantarray Minis.
Awesome. Thank you, Christos. Okay, next question. Can I deviate from your recommended protocols?
So
I
well,
think
the
Christos
answer
and
is
I are
yes.
ready to say, no, don't do it.
Yeah, no, absolutely. So we do have the recommended protocol. So everything we've shown so far, and we share with you some protocols when you get on board with our technology. But you have the freedom to cast whatever you would like. Of course, you'll have to run a study to make sure that what you cast will be functional at the end of the day. But the consumables and the technology is agnostic to the content of the tissues. As long as the tissue is healthy, then that is great.
Yeah, and I would just follow up with that by saying, if you're brand new to 3D tissue engineering, using our system, especially as we would recommend that you follow our protocols the first few times until you get used to the system and you have success in making your engineered muscle tissues. It's really quite, when I'm training new employees, we can go through casting one time and then they are up and running. They're making their own tissues with a very high success rate. So the platform and making these tissues is quite easy. But I would say, if you're new to it, follow our protocols the first time, and then you can get creative and start, incorporating different cell types and whatnot to make your own engineered tissues. I would also like to go back to that disease modeling question for 1 second. So I would like to clarify that, a lot of our calcium data that I showed you we have looked at in some of our disease models, and we do see, a clear disease phenotype presenting in some areas, calcium handling being one of them in our different disease models in both the standard or the classic size tissues and the minis consistently. So we do have data that is pointing that disease modeling is reproducible in both size tissues.
Awesome. Thank you both. Okay, next question is from Hadar. Which skeletal muscle diseases can be modeled using the mini Mantarray?
I think probably just about any disease model can be used. The bigger question is whether or not you're going to see a disease phenotype with your model. And that's always a question that we have when we're developing new models in-house, is will that disease phenotype present in vitro? And I think a lot of times to see a clear phenotype, you need to have a more mature or a well-developed muscle construct. And so that's one of the things that we're always trying to achieve with our platforms, is to produce, highly functioning muscle that is representative of what you would find in vivo. So, I think you can try any model. So we have a variety of different models in skeletal muscle and cardiac diseases. XLMTM, DMD, DM1 is a brand new model that we're going to be releasing here very soon, and we're very excited about that. The data looks amazing. So really, I think the sky's the limit. So you can try any disease model in our platform and see if your phenotype presents and it produces the data that you need for your experiments.
Yeah, and I would like to emphasize something, adding to Shawn's answer, that sometimes it's not necessarily a limitation of the consumables, the instruments and whatnot, is that the resources for modeling these diseases just don't exist. And this is exactly what Shawn is saying. We are providing some new cell lines, some additional media to be able to model these diseases in vitro, right?
Okay, thank you both. Our next question is, do you sell iPSC-derived cardiomyocytes?
I can answer that. So we have not commercialized any iPSC-derived cardios yet. However, we can provide those cells to you using healthy and different disease models through service projects at the moment. So you can reach out to us if you have a particular cell type that you need for your studies, and we could potentially get that to you through a service project.
Okay, great. Thank you, Shawn. Next question. What is the calcium concentration in your cardiac media?
It's a great question. So it is 1.8 millimolar, which is physiologically relevant. And one of the main goals of developing our formula was really to try to replicate many of the metabolites that you find in vivo in serum concentrations in vivo. And so we really try to keep a lot of the factors in our formula at physiological concentrations. Calcium is a big one that is a concern for many cardiac scientists. And so that is at physiologically relevant concentrations at 1.8 millimolar.
Perfect. Thank you, Shawn. Next question is, how is your cardiac media influencing maturation of the muscle?
Oh boy, that's a great question. And, that's not an easy answer. So there's a lot of different factors that we look at, or that we can look at when we're thinking about maturation of these cardiac tissues. A lot of the data that I showed you is some of the functional things that we look at right off the bat. So we're looking at, force and kinetics and calcium handling, which can speak to different kinds of ion channels that might be present, the expression level of those ion channels. We look at functional metrics like positive force frequency, positive post-rest potentiation. Those are always the more easy things to see in our platforms. But we want to do more extensive characterization, and we probably need to do more extensive characterization of our muscle tissues to really speak to the level of maturation that we're seeing. And that includes, probing, looking at protein expression of different myosin isoforms and different ion channels, structural organization. So looking at the sarcomere, we want to know, do we see more dense sarcomeres in our cells? Are the sarcomere length longer with our muscle bundles? So I think there's a lot of different things that we can look at, electrophysiology and metabolic function. And so our characterization of the true maturation state of the muscle that we're producing, I think, is not completely characterized yet, but those are definitely things that we're working on and trying to find, more definitive data that points to the physiological responses of the tissues that we're producing in our system.
Thank you, Shawn. Next question is, have you seen positive force frequency with the media?
Yeah, that's another great question. So that's something that we're currently working on, and the short answer is yes, we are seeing positive force frequency. So we're seeing it up to about 2.25 hertz. So this is another thing that we haven't well characterized yet, and we have more questions and exactly what's driving that response. We have a lot of different factors that we're applying to the muscle, both mechanically and through the media. And so we really want to have a good handle on what's driving the response that we see in our muscle bundles. And so hopefully within the next few months or so, we'll be able to provide you data that shows how our media might be playing into that effect, as well as other components that might be driving that response. But yeah, so far we are seeing some positive results for positive force frequency.
Fantastic. Okay, thank you both for all those answers, and thank you everyone for sending in all those great questions. We have now reached the end of our Q&A portion. If we didn't get to answer your question here, or you have questions that come up later on, please don't hesitate to connect with us via email at contact@curibio.com, and we would love to make sure those questions get answers. A big thank you to everyone for participating in today's webinar. You will be receiving a follow-up email from us with access to a recording of the event. And then, of course, another big thank you to both of our speakers, Dr. Shawn Luttrell and Dr. Christos Michas, for that wonderful presentation. We hope you found this webinar informative. Have a great day, everyone.
Thank you for attending, everyone.
Thank you.